All right, everybody. Hope you're doing well. Welcome to another Fix Your Gut live stream. I'm your host, John Brisson. Um, just want to let you guys know that we are updating Fix Your Gut. You can come here and download the Optimal Health Blueprint, which is recommendations for general supplement stacks for both men and women. All you have to do is provide your email, and then, of course, our blog is now been changed to research tab up at the top. And here we have some more blogs being released by Jason Hooper. We should have some of my blogs released in the next few weeks. And of course, we also offer coaching as well, health coaching from me if you have any gut issues or health issues or need supplement recommendations. If you need any diet or lifestyle or fitness coaching, of course, contact Hooper. Extremely knowledgeable. Anything with biohacking, I'm a little bit more conservative than Hooper is. But yes. So, today's video is going to be on are there any microbiome differences between a cesarean section and having a natural vaginal delivery? And there are differences. That is for sure. So why don't I begin? Stunted microbiome and opportunistic pathogen colonization in cesarean section birth. This was a paper published in October 2019 in the journal Nature. Immediately after birth, newborn babies experience rapid colonization by microorganisms from their mothers and their surrounding environment. Diseases in childhood and later in life are potentially mediated through perturbation of the infant gut microbiota colonizations. However, the impact of modern clinical practices such as cesarean dissection delivery and antibiotic usage on the earliest stages of gut microbiota acquisition and development during the neonatal period, less than 1 month, equal to 1 month, remains controversial. Here we report disrupted maternal transmission of bacteroids. Well, now bacteroids are gram-negative bacteria that are very important for your body to learn the gut-associated lymphatic tissue of your immune system to develop early in life. Even though they're gram-negative, they tend to be more opportunistic and less pathogenic. Average person has about 20 to 30% of bacteroids making up their entire intestinal microbiota. However, if you have too many, it can cause issues because they are gram-negative. They're endotoxins. It can cause inflammation in the gastrointestinal tract. They could also start eating the lining of the gastrointestinal tract and ferment protein and cause inflammation. Many people do have bacteroides dysbiosis that have coached. However, they are, probiotic or opportunistic. They're not pathogenic primarily. Like you would think of some bacteria that seem to only cause disease or are more likely to cause disease, like H. pylori, for example, or Legionella, which is the cause of Legionnaire's disease, or Klebsiella, which is the cause of rheumatoid arthritis and alkalinosing spondylitis. And a higher level colonization by healthcare-associated opportunistic pathogens, including Enterococcus, Enterobacter, and Klebsiella species, and babies delivered by cesarean section, and to a lesser extent in those delivered vaginally with maternal antibiotic prophylaxis or not breastfed during the neonatal period. Applying longitudinal sampling and whole genome shotgun metagenomic analysis of 1,679 gut microbiotas of 771 full-term UK hospital-born babies and mothers, we demonstrate that the mode of delivery is a significant factor impacting gut microbiota composition during the neonatal period. The persistence to infancy, 1 month, 1 year, matched large-scale culturing and whole genome sequencing. Over 800 bacterial strains cultured from these babies identified virulence factors and clinically relevant antimicrobial resistance and opportunistic pathogens that may predispose to opportunistic infections. Our findings highlight the critical early roles of the environment, mother, and hospital in establishing the gut microbiota in very early life. It identifies colonization with AMR carrying healthcare-associated opportunistic pathogens as a previously unappreciated risk factor. The acquisition and development of the early life gut microbiota follows successful waves of microbial experiences and colonization that shapes a longer term microbiota composition and function. Early life events, including cesarean section delivery, formula feeding, and antibiotic exposure that could perturb the gut microbiota composition, are associated with development of childhood asthma and atopy. While recent studies have provided substantial insights into the gut microbiota development during the first 3 years of life, many were limited by taxonomic resolution provided by 16S RNA gene profiling. Small sample size or limited sampling during the first month of life, neonatal period. High-resolution metagenetic studies of large longitudinal cohorts are required to establish the impacts and risk of early life events on the gut microbiota assembly, particularly during the neonatal period, while pioneering microbes could influence subsequent microbiota and immune system development. They characterize the trajectory of the gut microbiota acquisition and development during the neonatal period. We enrolled 596 healthy term babies through the baby biome study. Fecal samples were collected from all babies at least once during the neonatal period, less than 1 month, with 302 babies resampled later in infancy. Maternal fecal samples were also obtained from 175 mothers paired with 178 babies. Metagenomic analysis from 1,679 fecal samples from 771 babies and mothers revealed temporal dynamics of the gut microbiota development and increased diversity with age. Strikingly, the gut microbiomes exhibit substantial heterogeneity inter-individual and instability intra-individual during the first weeks of life. Individual differences explain 57% of the microbial taxonomic variation, followed by the sampling age of 5.7% of the variance. These results indicate the gut microbiomes were highly dynamic and individualized during the neonatal period, even more than observed in infancy. To determine the impact of the clinical covariates on the composition of the gut microbial community, we have performed cross-sectional permanova stratified by age. Mode of delivery was most significant factor driving gut microbiota variation during the neonatal period. Of our clinical covariates associated with the hospital birth, perinatal antibiotics, duration of hospital stay, and breastfeeding exhibited similar effects. Largest effective delivery mode was observed on day 4, with dissipated with age, but remained significant at the point of infancy sampling. No difference was observed in maternal gut microbiotas by delivery methods or neonatal gut microbiotas by elective emergency C-section births. So here we have, and the C-section microbiotas do change to look more like vaginal birth within days to some degree. But what effects can that have on a person's health? We know that C-section babies are more likely to get asthma. So here we have, vaginal birth having higher gram-positive bifidobacterium bacteria, which is a probiotic bacteria. It lives primarily in the colon. And also having a little bit more E. coli, which is a gram-negative bacteria. Is a proteobacteria. There are some species of opportunistic and there's some species of pathogenic E. coli. And then we have bacteroids being higher too as well. We have less Enterococcus, less Streptococcus, less Staphylococcus, and less Klebsiella in the vaginal births than we do the C-section, especially on day 4 of the C-section. I mean, there was a huge relative abundance of Enterococcus, a huge relative abundance of Streptococcus, a decent relative abundance of Staphylococcus, a good amount of Klebsiella, which Klebsiella actually increases as the baby gets older. A lot of strep and staph, a lot of those bacteria live on one's hands. Now, granted, the doctors who are performing cesarean sections are wearing gloves, but since there's just so much embedded in the environment of our skin, it would make sense that since the baby does not get the vaginal seeding of going through the birth canal, instead they pick up whatever, even though the doctors are wearing gloves. There's still enough strap and staph on those gloves in the environment that isn't through the vaginal birthing canal to inoculate the babies with infants with a good amount of opportunistic or infectious bacteria like Streptococcus, Staphylococcus, staph infection, strep infection, Klebsiella, which my belief is the cause of alkalizing spondylitis and rheumatoid arthritis. And people with certain gene mutations that allow their body to be more susceptible to colonization and infection of Klebsiella, or maybe more susceptible for their endotoxins. They are gram-negative bacteria, proteobacteria, to inflame the body's joints or the spine, the human body. So this is, again, quite concerning if you have a C-section. The differences in between the infant's microbiotas. Given the significant effect of the mode of delivery during the neonatal period, we next sought to understand how the microbiota composition development trajectory were altered. Samples from babies delivered vaginally were enriched with Bifidobacterium, E. coli, and bacteroids, while the commensal genera, comprising 68.3% of neonatal gut microbial communities. So commensal meaning it could cause problems, but generally they don't. Like Bifidobacterium is a probiotic bacteria, in my opinion. It's not an opportunistic bacteria. It's more likely to have a benefit for the host than to cause an issue, which validated the recent observations in other cohorts. In contrast, the gut microbiome of C-section delivered babies were depleted of these commensal genera, and instead were dominated by Enterococcus, Enterococcus faecalis, and Enterococcus faecium. Both are opportunistic. Staphylococcus epidermis, Streptococcus parasanguinus, Klebsiella oxytosa, and Klebsiella pneumoniae, which you recognize pneumonia. So Klebsiella pneumoniae is a cause of pneumonia, bacterial pneumonia. Enterobacter cloci and Clostridium perfringens, which is can cause food poisoning, which are commonly associated with hospital environments and hospitalized preterm babies. Yep, these are bacteria that, hospitals, they use harsh chemicals to sterilize them, disinfectants. So these bacteria are able to survive disinfectants easier than some of your probiotic bacteria like Bifidobacterium or Lactobacillus are not as hardy. So these bacteria are in the environment. They're in the hospital environment, like I mentioned. They're on the gloves of the surgeons, whether they're try to scrub or clean their hands as much as possible. So therefore, they're given to baby. In hospital, which are commonly associated with hospital environments and hospitalized preterm babies. On day 4, species belonging to this genera accounted for 68.25% of the total microbiota composition in C-section delivered babies. Previous studies reported that compared to C-section delivered babies, the gut microbiotas of vaginally delivered babies were enriched with lactobacilli associated with the mother's vaginal microbiota. However, we observed no statistic difference in prevalence, vaginal 11.9% versus C-section 15.7%, present at over 1% abundance of lactobacillus between vaginally or C-section delivered babies. Okay, that's good. So yes, the vagina is home to many species of lactobacillus, which is a gram-positive Firmicutes bacteria. It's also a probiotic bacteria. It produces lactic acid, which changes the pH, decreases the pH of your intestinal tract and decreases the pH of the vagina. So yeah, it's interesting that there wasn't much of a difference. You expect that there would be in previous, studies. You need more studies to replicate that, obviously. Some studies have shown that C-section babies probably do have less lactobacillus, but it's good to know it may not be as bad as we previously thought. Rather, commensal species from the bacteroid genus were detected at high abundance in the gut microbiota of 49% of vaginally delivered babies. In contrast, bacteroid species were lower, absent in 99.6% of C-section delivered babies. This low bacteroids profile persisted into infancy when bacteroids became the only differentially abundant species between vaginally and C-section delivered babies. Bacteroids are very important for priming one's immune system. In the gut, the gut associated lymphatic tissue. Although we could not access the independent effect of maternal antibiotic exposure during C-section delivery, as antibiotics were administered in all C-section deliveries among vaginal delivered babies, we observed a statistically significant association between the low bacteroids profile with maternal intrapartum antibiotic prophylaxis, which also accounted for the greatest amount of gut microbiota variation in vaginally delivered babies. Results expand on previous findings and further highlight a low bacteroids profile as a perturbation signature associated with C-section and maternal IAP in vaginal delivery. So if you're having vaginal delivery and you're giving prophylactic antibiotics, your baby will also have less bacteroids as well. Remember that. It's interesting. Material transmission of gastrointestinal bacteria to their babies is an underappreciated form of kinship. To assess if the neonatal microbiome variation can be attributed to different transmission of maternal microbiota, referred to bacterial strain transmission across 178 mother-baby dyads. We show the majority of maternal strain transmissions during the neonatal period occurred in vaginally delivered babies, and a much higher frequency in comparison with those of C-section, from 74.39% to 12.56%. Bacteroid subspecies, parabacteroid subspecies, E. coli, and bifidobacterium species were most frequently transmitted from mothers to babies through vaginal birth in agreement with previous observation in smaller cohorts. I meant to say bacteroid species, not bacteria subspecies. Bacteroid species, parabacteroid species, and bifidobacterium species. For bacteroid species such as B. vulgaris, the lack of transmission continued far beyond the neonatal period in C-section born babies. The late transmission of Bacteroides vulgaris rarely detected later in infancy. Wow. This is a contrast to the transmission pattern of other common early colonizers such as Bifidobacterium longum and E. coli, for which colonizations of maternal strains occurred more frequently later in infancy. The result highlights the neonatal period as a critical early window of maternal transmission, with disrupted transmission of pioneering bacteroid species evident in C-section babies with long-term bacteroids absence. So that's going to have a major effect on a person's development of their innate immune system and their digestive tract. While C-section babies were deprived of maternal transmitted commensal bacteria, they had a substantially higher relative abundance of opportunistic pathogens commonly associated with the healthcare environment. These enriched species included Enterococcus faecalis, Enterococcus faecium, Enterococcus cloacae, Klebsiella pneumoniae, Klebsiella oxytosa, and Clostridium perfringens, some of which are members of the ESKEPE pathogens responsible for a majority of non-nosocomial infections. So majority of your hospital infections in a hospital environment, healthcare environment, come from these pathogens. Indeed, the frequent gut microbiota colonization in C-section newborns was underreported in previous smaller cohorts, with insufficient statistical power. Among C-section born babies, 83.7% of opportunistic pathogen species during the neonatal period, as in comparison to 49.4% of the vaginally born babies. During the first 21 days of life, the healthcare-associated opportunistic pathogens accounted for 30.4% of the species-level abundance in the gut microbiota of C-section babies. Wow, compared to 9.8% in the vaginal babies, with the greatest difference observed on day 4. Longitudinally, the differences in combined opportunistic pathogen abundance persisted in the C-section babies who sampled later in infancy. C-section 2.8% versus vaginal 1.6%. Increasingly frequent and abundant carriage of opportunistic pathogens were also observed in low bacteroids vaginally delivered babies. The absence of breastfeeding during the neonatal period was associated with a higher carriage of Clostridium perfringens, Klebsiella oxytosa, and Enterococcus faecalis. So yeah, if you have a C-section baby, you definitely want to make sure you breastfeed your baby if all possible. It may help prevent some of the issues with having dysbiosis within the microbiome. All right. So for some more information, C-section versus vaginal delivery, long-term infant outcomes, and the hygiene hypothesis. Okay. During vaginal delivery, the contact with the maternal vaginal intestinal flora is an important source of the start of the infant's colonization. During cesarean delivery, the direct contact is absent, and non-maternally derived environmental bacteria play a role in an infant's intestinal colonization. Some authors have suggested the composition of the very first human microbiota could have had long-lasting effects on the tested and breastfed infants. For example, the study Gronlund et al. Showed that primary gut flora in infants born by C-section may be disturbed for up to 6 months after birth. Another study using culture-based techniques showed the mode of delivery was associated with differences in intestinal microbes 7 years after delivery. Now, some of the more recent studies have shown, like the one I just read, it may not be that much of a difference after about a month or two. Maybe, especially if you breastfeed. The clinical relevance of the changes is unknown. Even longer follow-up is needed to establish how long-lasting these alterations of the primary gut flora can be. Nevertheless, there's accumulating evidence that intestinal bacteria play an important role in the postnatal development of the immune system. Thus, if the intestinal flora develops differently depending on the mode of delivery, the postnatal development of the immune system might be also different. Available epidemiological data show that atopic diseases appear more often in infant after C-section delivery than after vaginal delivery. The composition of enteric microbiota in early days of life seems, therefore, to be a very important factor for achieving and maintaining good health in the years to come. It follows that it's fundamental to identify more thoroughly the intestinal ecosystem of the newborn. Although there is an increasing body of evidence that the intestinal microbiota play an essential role in the postnatal development of the immune system, the mechanisms remain poorly understood. Another study, Malamitsi Puchner et al., found that only vaginal delivery promotes the production of various cytokines implicated in neonatal immunity. Another study, Hallstrom et al., found a link between C-section delivery, disturbed intestinal colonization, and possibly occurrence of necrotizing enterocolitis in preterm infants. Although the epidemiological studies demonstrate that elective C-section delivery provides an increased risk for allergic diseases in later childhood, confounding factors could also play intermediate roles. Data available from several studies indicate a delayed onset of lactation with C-section. Thus, many infants born by C-section also lack the early support of breast milk as a stimulator for a physiological intestinal flora. Both the non-physiological start of colonization and the missing early dietary support by delayed start of lactation may result in these long-lasting effects. Yes, if you're going to have an emergency C-section or you know you're going to have a C-section, if you know you have a C-section, definitely ask your doctor about supplementing with fenugreek, if all possible, before or even after the C-section, because fenugreek as an herb does improve lactation. It does improve milk production. And side benefit, it makes your armpits smell like maple syrup. But yes, that could help. My wife was able to breastfeed immediately. We had an emergency C-section with Holden, my firstborn. We had a C-section had to be done with Abel because of his complex medical condition. And my daughter, Amelia, was also delivered by C-section too as well. But my wife didn't have any problems producing milk. And they were breastfed for 2 years, each of my children. Babies are born with immunological tolerance that is instructed by the mother by preferential introduction of regulatory T lymphocytes, which may allow the baby to become colonized by this first inoculum. The mechanism is via substantial numbers of maternal cells crossing the placenta to reside in fetal lymph nodes, inducing development of CD4 plus CD25 hyphox P3 plus TREGs that suppress fetal and anti-maternal immunity. Those are immune cells, by the way. And persist at least until early adulthood. However, only a subset, if any, of these microbes to which the newborn is initially exposed will permanently colonize available niches and contribute to the distinctive microbiota harbored by the body habitats of adults. As more and more deliveries bypass the vagina, babies might not be exposed to these microbes at birth. Differences in delivery mode have been linked with differences in the testable microbiota of babies, multiple studies. Intentional communities may serve as a direct source of protective of pathogenic bacteria very early in life. Another recent study offers a detailed look at the early stages of body's colonization by microbes. Babies born vaginally were colonized predominantly by lactobacillus, whereas C-section delivery babies were colonized by a mixture of potentially pathogenic bacteria, typically found on the skin and in hospitals, such as Staphylococcus and Acinetobacter. Suggested babies born by cesarean section were colonized with skin flora in the lieu of traditional vaginal type of bacterium. Now, the study we just saw said that maybe lactobacillus might not be much difference between C-section babies and vaginal delivered births. But just like I mentioned earlier, what we're finding in these babies that are born by C-section are microbes that would normally be found on one's hands, and microbes that would normally be found in a hospital, that are extremely pathogenic, by the way. The effect of the mode of delivery on development of childhood disease has just recently begun to be explored. The effect appears to be more robust in areas of immune-mediated diseases. C-section delivery has been associated with significant increased rate of asthma, especially in females, and allergic rhinitis, but not atopic dermatitis. This increase was even more apparent when counting for the factors surrounding the cesarean delivery. The risk of asthma was increased by 60% in females who underwent a repeat cesarean without ruptured membranes versus those babies with ruptured membranes or labor prior to cesarean delivery. So here we have increase of allergic rhinitis. Repeat C-sections, there's more likely chance for allergic rhinitis, which is pretty much having hay fever, allergies in your nose. Asthma, also cesareans. If you're female, born higher. Female repeat, even higher. Celiac disease, almost as high as female repeat cesarean. Diabetes, slight chance elevated. Gastroenteritis, greater chance. Gastroenteritis and asthma, 1.74, pretty good chance. children born by cesarean, and it might be because there seems to be there's studies between people with celiac disease or people with gluten insensitivity having less reactions to gluten or not even having reactions to gluten once they have a good amount of bifidobacterium in their colon, which seems to render the gliadin and glutenin proteins that make up gluten um, less problematic to the human body. And we saw in the previous study, C-section delivered babies have less bifidobacterium by quite a substantial amount. No association was found between cesarean delivery and Crohn's disease and ulcerative colitis. However, while preterm birth has been implicated in development of inflammatory bowel disease, motor delivery has not. Yeah, and again, I think it's Crohn's disease and ulcerative colitis are caused by microbacterium avian impaired tuberculosis. I've coached many people into remission just by having them avoid ruminant animal products. You, of course, you have to take some supplements and lifestyle changes and everything, but that does seem to help. Seems to be more genetic in nature. Crohn's disease and ulcerative colitis does, of whether or not a person, because MAP is in our environment. Most people, it enters their digestive system, but there's certain genes. If they don't have those gene polymorphisms or mutations, or if their health is good, if their diet is good, the microbiome is good, then the Microbacterium avian impaired tuberculosis doesn't contribute to developing of inflammatory bowel disease. So we have another study here, more recent, 2020. Cesarean delivery and risk of chronic inflammatory diseases. Population-based registry study of 2,699,479 births in Denmark during 1973 and 2016. So again, we have more data here showing that C-section births put you at a greater risk for diabetes, a greater risk for arthritis, a greater risk for celiac disease, which seems to be one of the bigger risks. Somewhat greater risk for inflammatory bowel disease, but not as much. So it seems to be the greatest two, again, being confirmed by the study, is C-section will give you a greater chance of developing asthma, and a greater chance of developing celiac disease. So what can be done? Okay, so first of all is breastfeeding. Breastfeeding as early and as soon as possible. Get that production of milk up as best that you can. That's crucial. There's something called vaginal seeding where they swab the mother's vagina, and they kind of take the swab and they put it around the baby's mouth and skin and stuff to kind of transfer the microbiome. But the studies are kind of mixed. I don't really recommend it until more studies come out. There is another thing I'm about to mention earlier, which is to give a fecal matter transplant to the healthy mother to the baby. I think that could be done. That's something that needs to be studied too as well. As many people who have had children, that you tend to have a bowel movement while you're giving birth. So babies do get inoculated from the fecal microbiome. It says right here, indeed, anecdotal accounts suggest most women deliver babies vaginally do defecate during labor. Feces is always part of the delivery process, agrees obstetrician Bo Jacobson, the University of Gothenburg, who is not part of the research team. He says he therefore thinks that from a scientific point of view, the researchers have done the right thing. Even on first impression, the procedure seems nasty. So we need more studies on vaginal seeding. We need studies on mother fecal transplants to C-section babies. We do know that breastfeeding as soon as possible and early as possible is one thing that may help. Another thing would be to get this Smidge infant probiotic powder that we recommend on Fix Your Gut. And talking to your doctor, your obstetrician, and your baby's pediatrician of whether or not, giving a very small dosage to your infant who's been delivered by C-section and mixing it hopefully into breast milk to see if that will also help their microbiome. There's no histamine producing strains in here. There's no D-lactate producing strains in here. Just got a few strains of bifidobacterium. It's good for the development of the baby's colon. Remember, C-section babies have lower levels of bifidobacterium, and 2 strains of lactobacillus. So yeah, I mean that would be another good thing that you can do. So far we have get the baby on breast milk as soon as possible. Their fenugreek may help with milk production. And there's also techniques that a certified breast milk consultant, midwife, might be able to help give you tips on how to massage your breasts properly, women, to improve milk production. So there's that. There's possible fecal matter transplant in the future. We need more studies. Vaginal seeding needs more studies, in my opinion. And we do have, you could add the infant probiotic smidge, just really small amounts to start out with your newborn. Probably of about, I would say, they give you the little spoon to do with. So I'd probably say a fourth of the little, very little tiny spoon, just a really smidge to start with. And to tolerate, you can go up to 1 full spoon, which is about 1.4 billion bacteria. That can also help, of course, run that by your healthcare professional as well. But yes, there are microbiome differences between C-section and natural deliveries. And the C-section delivered children have more commonly found opportunistic and pathogenic bacteria that are found in hospitals and found on skin, like Staphylococcus, Streptococcus, Klebsiella, Enterobacter, and have lower levels of probiotic E. coli or opportunistic E. coli, probiotic bifidobacterium, probiotic or opportunistic bacteroid species, and so in doing so, it does affect their microbiome. However, through breastfeeding, through maybe using the smidge infant probiotic as possible, through fecal matter transplants when they're more studied and become available in the future, maybe those differences can be eventually lessened, so that there is not much of a statistical difference between a baby born by C-section, where the mother, does these things, or compared to them not doing them today. All right, everybody, take care. See you soon.